All right. Good morning, everyone. Thanks for tuning in for the ISHR MCI webinar today, where Min and Ionis will be presenting, and Obed and Andrew are going to be moderating these talks. Just a few housekeeping items to go over from the MCI committee before we get started. I want to introduce everybody. So these are the mugshots of everybody on the MCI committee. So this committee ranges from senior assistant professors to newly promoted full professors who are typically 8 or more years beyond their first faculty appointment. And what this committee does, so we planned the mid-career symposia at the ISHR meetings. We plan things like this webinar. And so this webinar happens quarterly. And so if anybody listening considers themselves an MCI and they want to get involved and volunteer either to be part of the committee or part of the webinar, please reach out. We're seeking volunteers for both of those things. Reminder that the ISHR North American section website is live, so you can find more information about the committee, the upcoming meetings, and renew your membership or join here via the website. Thank you to everybody who was able to attend the ISHR World Congress in Nara last month. It was a lot of travel, a lot of jet lag, but a good time for those who were there. And the meeting next year will take place from May 30th to June 4th in Minneapolis. And the co-chairs for this meeting are going to be Tim O'Connell, who is a member of the MCI committee, Jaap van Berlo, and Julia Liu. And the MCI we will be sponsoring 2 awards at this meeting. The first is the MCI Leadership Award. So for the Leadership Award, there will be 3 finalists who will present at the meeting. And the Leadership Award will recognize an outstanding mid-career scientist who is making major and independent contributions to the advancement of cardiovascular science and leading a growing research program who is likely to play a significant role in the future. The Leadership Award will present a major lecture, receive an honorarium and a plaque. And I also want to point out that the nominations for this award, so you can self-nominate for the Scholarship Award, but you need to be nominated by someone else for the Leadership Award. And so if anybody has any questions about either of these, feel free to reach out to Kelly or anyone on the MCI committee, and we can make sure your questions get answered. And with that, I think I'm going to turn it over to Obed to make the first introduction for Min, our first speaker.
Hi, everybody. Good morning, wherever you are. My name is Obed. I work with Dr. Kika Sugarov in the University of Colorado Anschutz. It is my pleasure today to introduce today's first speaker, Dr. Min Shi. Dr. Shi is an associate professor of medicine at the University of Alabama at Birmingham, where he leads an active research group focusing on developing novel therapies for patients with acute myocardial infarction or with ischemic perfusion injury like we are seeing. And Dr. Shi began his career with a strong foundation in both clinical training and basic research. He matched his internal medicine residency at UT Southwestern and went on to complete a prestigious cardiology fellowship over there. During his fellowship, he studied cardioprotective effects of HDAC inhibitors on ischemia perfusion injuries. His work using the FDA-approved HDAC inhibitor SAHA demonstrated a 40% reduction in infarct size in large animal models and earned him multiple awards and a publication in Circulation. Following this, Dr. Shi pursued mechanistic studies using novel autophagy-inducing agents and cardiac-specific knockout models, leading to a KO8 Mentored Physician Scientist Award. Now at UAB, his research, supported by NIH and AHA grants, focuses on mitochondrial stabilization and autophagy regulation to mitigate cardiac ischemia reperfusion injury. He recently received a Department of Medicine Research Award at UAB for maintaining full-time research support and has mentored several graduate students and postdoc fellows. Dr. Shi is a dedicated physician scientist whose work exemplifies the power of translational research to improve outcomes in cardiovascular disease. Please welcome me and welcome in Dr. Min Shi. Thank you.
Thank you, Obed, for this generous, introduction. I also thank the ISHR, MCI committee for the invitation. So I will discuss some the background of this cardiac ischemia perfusion injury and the current status art, how to treat that. Then I will give some information on what's my research contributed to this field and the future direction. So cardiovascular disease still the leading cause of death whole world, but there is a little bit twist in 2020. The Lancet published, in high-income countries, the cancer costs twice as much as death as cardiovascular disease. The major reason is that we're doing a great job to reducing this mortality rate in cardiovascular disease in developed country, with the development of this revascularization and the novel therapy for this aortic stenosis and the popular use of statins all contribute to this. However, in the whole country, in the whole world, this cardiovascular disease still causes the most death. One of the most common cause is this acute myocardial infarction. We call it ST elevation myocardial infarction. And this is the EKG when the patient came to the ER, or even in the ambulance, they can do a EKG. You see the ST elevation at 1 AVL, and also from V1 all the way to V4. And when you see this, you rush this patient to the ER and also notify the hospital that has a left heart cath facility. So they put a catheter in and inject dye, and you can see the circumflexus opens pretty well from the left main, but there's a abrupt occlusion of the LAD, left anterior descending branch of that. That's a widowmaker. It's a complete occlusion, and then what we do is we put a guide wire and put a stent in there. You can see all of this blood flow has been restored. This is the key. Without revascularization, all of these cells will die. And there are 1.5 million new cases of acute myocardial infarction, including the non-ST elevation, myocardial infarction. So the infarct size for the STEMI is a major determinant of onset of heart failure. If the infarct size is large, the heart will fail faster. And so reducing infarct size is a major clinical interest. There are several ways to do that. And let me introduce you to this ischemia reperfusion injury. When the coronary artery is occluded, the infarct size will increase and with time. And with reperfusion, with the stent, you can save some of the myocardium. You can reduce the infarct size pretty significantly. However, with a lot of studies showing, even with this opening of the occluded coronary artery, the cell death continues increase. And if we can block this reperfusion injury, we can save another like 25% of that of this infarct size. Here is a landmark for the STEMI therapy, and it's starting with the 1970s. For the ischemia injury, we know we have done a lot of important study. So if you don't reperfuse, the necrosis will be almost near complete for the perfusion area, around like 90%. And then if you open it up by thrombolysis, or when later they did also says angioplasty, the PCI and the stents, they can reduce infarct size. Then they have all of this, antiplatelets. It's pretty well studied. And up to the 2020s, this reduced ischemia injury is already reached the maximum. We cannot reduce this infarct size further. In the late 1980s, and a lot of studies showed that preconditioning can reduce infarct size further. on top of when you open up the coronary artery. So the first evidence of reperfusion injury, you still have some cells can be saved. And then they do the remote ischemia conditioning and the post-conditioning. All of this has been done, in the small clinical trial, which works really well. And of course, they first tested in animal, then they went to the small clinical trial. It works really well. However, until now, we still don't have any standard therapy yet. And this is published 2015. And at that time, there are several studies use cephalosporin, to block calcineurin to reduce the MTTP, the mitochondria, transport opening. And then they did this preconditioning, and basically, before the reperfusion, they put the balloon up and down to delay the reperfusion. They also did a beta blocker to reduce the cardiac contractility, to reduce this oxygen consumption. And they also have the post-conditioning. It's just after patient goes in, after they put a stent, they can cause some like ischemia of the limbs. These all failed, unfortunately, in a large clinical trial. In a small clinical trial, they have like more selected population and more controlled condition. But when you go into the large clinical trial, they did not do that. It did not pan out very well. And why do we why do we care about this? And this is 2015. They did a cephalosporin with compared with control on top of the state of art revascularization, the reperfusion techniques like stents, this kind of thing. And although there is no difference between the control and the cephalosporin, but with like 12 months follow-up, there's a clinical outcome of death or heart failure is still around like a 24, 23%. So it shows how important to reduce this more injury, to prevent this death and heart failure in the long term. So because it's hard, and we're still trying, and there's lots of animal studies shows lots of gene or some treatment can work, and we're still trying. We were trying up to like 2024. There are several clinical trials going on, and you can do the infusion of a super saturated oxygen therapy, and they did like a small clinical trial works, and then we need to validate that. Then we have the Impella. Impella is like a left ventricular assist device. You can insert through the aorta and unload the heart. So they are doing this, and they're still ongoing, and they should should be reported soon. They are also doing this coronary sinus occlusion, so they reduce the reperfusion, flow rate. And then they also recognize this, the immune response is very important. And they use a CRP phrases to get rid of CRP to see whether they reduce the infarct size. So that all works in a small clinical trial. And you see the patient number like from 45 to 100. But it still have to be proven in a large clinical trial. Then what's a major mediators for this lethal reperfusion injury? And it has been studied a long time, and there's a good review showing this during the ischemia, and this is lower pH, and they cause this lactate accumulation. They can damage the mitochondria, and they also have the calcium overload. When the blood flow restored, then we get rid of the lactate and the restoration of the physiological pH and calcium overload. They can contract a lot. When they contract, they need a lot of energy, and then the mitochondria need to work extra hard. then it causes this damage to the mitochondria and generate ROS, then cause the cell death. That's an oversimplified view, but that's exactly what happened. And after the reperfusion, even within like hours, you can activate, the immune system. The first responder is the neutrophil, and then the macrophage, and then the T cells, and to form the scar. And so the mitochondria obviously is one of the center of that, and a lot of smart people get together and publish the review in 2017, and it's a Nature Review Cardiology. They have a look of this cardiac protection, which is like reperfusion injury, and they put all of this involved processes: autophagy, electron transport system, mitochondria permeability transition. This is the pore opening, peroptosis, and the inhalation anesthetics, and also the no flow phenomenon. And they all point to this mitochondria, because mitochondria is the first need to generate this ATP, and also handling with extra oxygen overload. So there's a dilemma treating the cardiac IR injury, and the more I study that, I figure out this is very important, and this what my understanding. While you have a heart attack, and then you first want to maintain the cardiac output. If the cardiac output drops, if the infarct size is too big, the patient will die very quickly. You don't have a chance. And then you can open this occluded coronary artery, and then you hope all of this native mechanism will repair the mitochondria, repair the damaged cardiomyocytes. Then you can let the patient get can survive better, and then reduce infarct size. There's a lot of things worked. Hypothermia works, and it works really well. And in the clinic, we cannot do that during. Why is the time limit? Another thing is they can limit the cardiac output. Then the heart work with very efficiently. However, with this like a smaller demand of oxygen and the cardiac output, you can save more cardiomyocytes for sure. So they can lay back and repair itself. Beta blocker works. You can give a lot of beta blocker to that, and they can reduce the oxygen consumption. At the same time, they can reduce the heart rate and reduce the contractility, then reduce the cardiac output. Impella also works. Impella is just unload the heart completely, and it's very costly. Then tens of thousands of dollars, and then have a procedure complication. And you really want to find something that can surprise this oxygen requirement, preferably to the ischemic, the risk area risk. And at the same time, you don't impact, the other, myocardium, and which can still support the cardiac output. So in short, it is that we have to maintain the cardiac output. At the same time, we can rest this myocardium at risk, so they can repair themselves. Okay. Then, I joined Dr. Hill's lab, 2010, and we did this study with HDAC inhibitor. HDAC inhibitor has been widely studied, in the cancer biology. It can regulate lots of, signaling pathways. Mainly, they can, if they give this HDAC inhibitor, they can amplify the cell signaling, and can possibly reduce this injury after reperfusion. SAHA is FDA approved for lymphoma, and we since it has been reported it can reduce infarct size in the mice. We tried in the rabbit, so trying to bring this to the clinical trial, and We have a pre-treatment and a reperfusion only treatment. And the pre-treatment, we give many doses, 4 doses before before the reperfusion. Reperfusion only is we give this at the reperfusion. This is very important because we cannot predict when the heart attack will happen. So that will give you a therapy, window. So the pre-treatment reduce infarct size and reperfusion only also reduce infarct size. And we did this echocardiogram. We found this can restore some of this cardiac function, partially restore cardiac function, in 24 hours. And it works so well. And what's the mechanism? We have this reporter mice for the autophagy. And this is LC3, which is autophagy, related protein, and labeled with RFP, GFP. And in autophagosome, its pH is neutral, so you will see a yellow dot. And then when it fused with the lysosome, pH will go down, and the GFP will be quenched, so you have autolysosome. If the autophagy flux increase, you will see the increase both a red dot and the green dot, and a yellow dot, sorry. And then that's exactly what happened with the SAHA, treatment at the border zone. You can see both the yellow dot and the red dot increased, compared with the DMSO as a control. So in my lab, when I started, at UAB, we want to look at what's SAHA's, effects on this mitochondria. So this is the EM, and the border zone, you see this compact, mitochondria, but with SAHA treatment, it's kind of relaxed, and we see the mitochondrial area increased, and both border zone. In the border zone compare with the DMSO, and with both pre and reperfusion only. We also found this mitochondria, at the border zone, the mitochondria protein level also increased. It's quantified here. And we look at it, the PGC1 alpha has been induced, at the border zone, and but not the PGC1 beta. And we also did the ATG7 knockout to remove this remove this autophagy, and I did not show that data here. So the summary of the finding of this SAHA, SAHA induced autophagy, we know that, and loss of ATG7 will abolish SAHA's cardiac protection. We showed in the circulation paper. And at the same time, they can induce PGC1 alpha, and loss of PGC1 alpha will prevent SAHA's beneficial effects on mitochondria. So all in all, it's autophagy and the PGC1 alpha, then the mitochondrial homeostasis. They can block the RS and reduce cell death. And what we think our most important contribution is this: when you have this reperfusion injury, there's a mitochondrial damage. Then the mitochondria will get release a lot of their contents. It's like exploding like a power plant. And autophagy will remove this damaged power plant with the mitochondria. Then at the same time, the PGC1 alpha will replace with new mitochondria that can support this energy, and the end result, it can reduce the cell death. Okay. And however, this SAHA has a lot of side effects. It's very toxic, to the animal even, and so it's very hard to bring to the clinical trial. Okay. And then we look, and we found that beta-hydroxybutyrate is the endogenous HDAC inhibitor. It's metabolized, with stress. The fatty acid will go into this liver and produce this beta-hydroxybutyrate. It can be used in the heart and the brain. And here are the other levels of this beta-hydroxybutyrate. And the therapeutic level is 1 to 3 millimole, and 2 days fasting or 90 minutes exercise can do that. SGLT2 inhibitor, which has been used in the diabetes, can also increase that. and extensive fasting is go to 6 to 8. Then the diabetic, ketoacidosis goes up to like a 20. So that's a level of this. This is detrimental, but this probably is good. So the beta-adjusted butyrate, the ketone body has a lot of, they can provide, energy, oxidative, oxidative energy generation, and then also combine the GPR and also inhibit HDAC and inhibit this inflammation. All of together will reduce this stress to the heart. And there are some reports showing this give this beta-adjusted butyrate before the IR, they can reduce infarct size in mice. So we want to test whether this beta-adjusted butyrate administered at the reperfusion can reduce IR, and whether it's affecting autophagy and mitochondria homeostasis. And we did a mouse IR, we did this 45 ischemia and gave a beta-adjusted butyrate. We got echo simulated IR in the cardiomyocytes. We gave 2-hour ischemia and 6-hour reperfusion to see the cell death, and we gave this. So we did this is mouse cardiomyocyte specific, and we also have access to acute myocardial infarction patients. At UAB we collect around 60 patients, and we measure the beta-adjusted butyrate at the baseline. And the baseline means, before the PCI, when the patient come in with a heart attack, we measure that. And after PCI, we measure that again at 24 hours. And they can reduce this, significantly. But it's only go up to like 0.5, probably not enough. So we did this process, give this beta-adjusted butyrate with 10 micromole per kg, and we measured this concentration of the beta-adjusted butyrate. They go up to around 6 at most, and by hour of like 3, it's almost back to the baseline. And we calculate the AUC, which is very important to guide a future clinical study and to see the drug exposure. And beta-adjusted butyrate has been given at a reperfusion, can reduce infarct size significantly, as good as SAHA. It's very good. And we did all of the ejection fraction, and after IR it goes down the ejection fraction, and with beta-adjusted butyrate we can maintain partially for 24 hours. This is very important because you want to maintain them. Again, you want to maintain the cardiac output. At the same time, you want to save this myocardium. And we did the same thing with this autophagy, and we found this beta-adjusted butyrate increase the red dot and also the yellow dot, and this shows it can induce autophagy. With data not shown, we showed that the mTOR pathway has been affected also. And then we did this mitochondria function analysis, and this TMRM is the mitochondria member potential. At baseline, the member potential has already improved with beta-adjusted butyrate, and with IR, this definitely protected the mitochondria member potential. And with the mito tracker to show the intact mitochondria is also increased with beta-adjusted butyrate. Then we looked at ROS. This is total cellular ROS, and IR increases ROS, and beta-adjusted butyrate will reduce a little. And then the mitoSOX is mitochondria specific, and we show the same thing. So the ROS has been blocked. And we also did a Seahorse bioenergetic measurement, and we see the proton leak all increased by this beta-adjusted butyrate, which reduced the RS production. So in summary, we found this IR injury caused mitochondrial damage and cell death, and the mediator is RS. And with beta-adjusted butyrate, we can induce autophagy. Autophagy will remove the damaged mitochondria, and at the same time, they can protect the mitochondria, and so reduce the RS and also maintain their function with their mitochondrial member potential. So this is possible a good therapy for this reperfusion injury. So I already mentioned what's the clinical, perspective. So beta hydroxybutyrate is stress metabolite during heart attack. We already showed that, and after reperfusion, with a stent, this beta hydroxybutyrate will go down. And one dose of this administered at the reperfusion can reduce this infarct size. Also, does not have much, toxic effect because it's physiological level. And beta hydroxybutyrate has been used safely in the heart failure patient. It may be a viable, therapy to reduce infarct size in the heart attack patient. And the future direction, we will test this in a large animal IR model, pig model, and this beta hydroxybutyrate is pretty cheap and will be a good therapy. And another view is to do this. To identify which HDAC isoform, SAHA and beta hydroxybutyrate are non-selective, HDAC inhibitor, and we are working on that. Hopefully, we can get more data in the future and give you update on that. And this study mainly done by Yuxin Chu and also Jing Yang, and with all support of our great staff, our animal surgeons and our great technique support. This is a whole lab, and we have like a Christmas party at that time. I also thank for NIH and AHA for this support. Okay, thanks you for your attention, and I will be happy to answer any questions in the chat.
Thank you so much, Dr. Shi. Please, if you have questions, please use the Q&A button to submit your questions, then I'll read it aloud for Dr. Shi. To start off, my question is, what do you think is the biggest translational barrier to moving SAHA and ketones to the clinic?
Yeah, that's a great question. SAHA, as I mentioned, is pretty toxic, and it caused a lot of problems, and we didn't even manage to reach that. And the biggest barrier is you want to have a safe drug, and you can give at the reperfusion, and also have a very good, protection efficacy, reducing infarct size like 50-60%. Then that can be translated into the large, patient population. Another thing is we all do this in the healthy animal, young animal. So it's easy to see any protection. And however, if we expand this to clinical trials, and we're seeing patients with diabetes in their 60s, 70s, and hypertension with multiple vessel disease. So hopefully, with as a high efficacy and a safe drug, they can be used on everyone. So we can see like a big decrease. Then we can see the mortality and heart failure, benefit. And then the mortality is hard outcome. Heart failure also hard outcome. You cannot fake any of that, and it's not intermediate. So that's a major barrier. And from the mice to the human, we need to do like a large animal. That's why we want to do a pig.
Okay, sure. Thank you. There's a question from Guampin Lee. They say great talk. Their question is that they are wondering if autophagy regulated by SAHA or ketones, specifically mitophagy. Okay.
Yeah, that's a great question. We have the MetaQC mice now, and we can test it, and we also have a parking, null and the parking active form. We are actively working on that. So with this SAHA and the beta hydroxybutyrate, I cannot say it's only mitophagy.
Okay. And then he followed up to say, did you further test like, other mitophagy agents?
Mitophagy agent. We did not do that. We have an active parking form, and there's a group already showed they target the parking activation, and actually it can reduce infarct size. So I'm
Okay.
kind of behind. So that has already been done, but mechanistically, whether we can still working on that.
Okay. There's a question from Limpiao. They say, nice talking with you. Which substrates did you use to measure OCR? And is it possible to measure OCR directly using ketone bodies?
Oh yeah, we measure the in the NRVM, and we did this the maximum respiration. I did not show the data, that's with a collaboration with so we measured the maximum and reserve, and also the proton leak ATP linked, and so in the NRVM, not in the adult cardiomyopathy, we still don't have any success with that. But that shows this the beta-hydroxy build rate, increase the ATP link, production, OCR, OCR, and also they suppress a little bit, the maximum, so they can reduce some of this oxygen consumption. The most important thing, they can increase the proton leak and to reduce RS.
Okay, sure. Thank you. And the next question is from Leslie Kennedy. They say, fantastic work. Have you looked at ketone treatment in mice with heart failure with treated or untreated diabetes to see if there's protection with diabetes?
Yeah, that's a good question. We did not do any heart failure study. The heart failure study is published in human, and they intentionally avoid the diabetes. And even I will, if I want to do this like a clinical trial, I don't want to include the diabetic, and we'll probably measure their ketone body level before we give anything. But this has been shown clearly in human. They gave beta-hydroxy butyrate infusion. They can increase their cardiac output. At the same time, reduce the vascular resistance. It's more like an inotrope, but without the bad effects of inotrope, like epinephrine and the butamine and mirinol. Yeah.
Okay, sure. Thank you. There's a question. The last one is from Rabia Anjum. They say, what do you think is likely the time window during which the ketone body therapy will be most effective?
Yeah.
And do you think it's going to be dose-dependent effect?
Yeah. That's a very good, question. There's a lot of study has shown, the ketone body level, the beta-hydroxy level, before they did fasting, all of that, and as I showed that in my review article, that's maybe like around 6. You don't want to go higher than 8 or something. That's cost damage. The time window, we only tried at the reperfusion, exactly at the reperfusion, but you can still extend, when the patient come in with a STEMI, you could give before the reperfusion. And the beauty of the HDAC inhibitor, and there's studies shown up to like 90 minutes after reperfusion, if you gave the HDAC inhibitor, can reduce infarct size as good as at that time. I did not do this delayed for beta-hydroxy butyrate because it's so easy to give, and but I expect this will be very clinically relevant.
Sure, thank you. I think that is the last question we have. Thank you so much for your time. And we're going to move on to the next presenter. Thank you.
Thank you.
All right, very great talk by Dr. Xie there. So our next speaker is Yanis Karakikis. He obtained his PhD in biological sciences with a focus in immunology at the University of Essex, and then followed that up with postdoctoral training with Dr. Roger Hajar, who is pretty well known for his expertise in gene therapy, and some of his work during his postdoc actually led to some of the first laid some of the groundwork for circuit 2 in human gene therapy heart failure trials. During this time, he was also awarded a K99 Transitional Award, which helped him to secure his first faculty position at Stanford University. where he has been since then, rising up through the ranks with a recent promotion to associate professor in 23. Currently, his research focus really lies and has continued to lie in the gene therapy approaches to treat cardiovascular disease, and his lab holds a significant interest in using iPSCs and human-engineered tissue organoids for mechanistic studies with the overall goal of refining precision medicine. Publications-wise, he's published extensively with over 80 peer-reviewed publications, 29 of those being lead or corresponding author. And several of these have been in top-tier journals, Cell, Nature, the European Heart Journal, and Circulation, where he also serves as a reviewer for several of these prestigious journals. He's been continuously funded through extramural support, serving as the PI of 5 awards with numerous others as co-PI. And in addition, he also has served as I mentioned, a top journal reviewer, but also as a chair for various AHA funding mechanism study sections. So without further ado, I don't want to take any more of his time here, but the ISHR MCI welcomes Dr. Karakakis and his presentation on serine metabolism and dilated cardiomyopathy.
Great. Thank you, Andrew, for the kind introduction. Let me share my screen here. You guys see my screen? Yes?
Yep, looks good.
Yeah. Can you hear me?
Yes.
Hi. Thank you, Andrew. Thank you again for the kind introduction and the invitation to share our research here with you today. And thank you all for joining and staying on Zoom for extending time. What I want to do today is I just want to walk you through our translational journey over the past 5 years of my lab's recent kind of efforts to develop some targeted therapies for dilated cardiomyopathy. I'll start with a very brief background on the clinical need and genetic complexity of dilated cardiomyopathy that we study extensively in my lab. First, I'll talk a little bit on our discovery path to using our IPS-based phenotypic screening approaches and the mechanistic dissection of the serine biosynthesis pathway. This is a study we published a couple of years, just kind of highlight the key findings of this study that led to the next part, which is the translational part. I'm going to spend a bit more time there, how we took this discovery towards a translation and proposing a new concept of metabolic reprogramming as a therapeutic strategy. And I'll show you some of our recent data on the preclinical validation of PGDH. This is a phosphoglycerin dehydrogenase gene therapy. And we should have time for questions and answer at the end. So by way of background, DCM is a very complex disease. It's a very complex disease, and the most common cardiomyopathy. It is characterized by this left ventricular dilatation and contractile deficit, and as the heart, as shown here, becomes dilated, becomes bigger and thinner, it's unable to pump blood and meet the energetic demands of the human body. In adults, its prevalence is estimated between 1 to 250 to 1 to 2,500. The age of diagnosis typically is between 20 and 50 years old. Etiology, half of it is considered genetic, and the other half is of unknown origins and called idiopathic. And currently, there's no disease-modifying therapy, and the only cure is heart transplantation. So there have been some significant progress over the past decades on understanding the genetic basis of the dilated cardiomyopathy, and the genetic architecture of the disease is very diverse, with more than 50 genes are being implicated or associated with disease pathogenesis. These genes are distributed across multiple cellular ontologies by chemical pathways, just shown here schematically. Genes in the sarcoplasmic reticulum, phospholamine, for example, right-arm receptor, a lot of genes in the sarcomere, troponin, tropomyosin, tidine, all of those have been implicated in the disease pathogenesis. There's the desmosome-related genes, nuclear genes like lamin A, one of the most common ones associated with the disease, and also some rare genes associated with cytoskeleton and extracellular matrix. And over the past few years, actually, there's been some large clinical genetic studies have provided direct clinical evidence that most of these genes do actually cause DCM. There's significant clinical evidence for about 18 genes. The others are still some association, but not causative. But despite these efforts, the underlying mechanisms remain elusive, translating to a lack of disease-modifying therapies. So my lab is interested in developing targeted therapies by understanding this genetic genotype to phenotype association. And our central hypothesis here is that if we can develop more effective therapies if we can link the mutation, the genotype to the actual phenotype and understand the underlying mechanism. And to test this hypothesis, we use a multidisciplinary approach that combines disease modeling using induced pluripotent stem cells with molecular and cellular phenotypic assays, functional assays, and ultimately my main interest is to develop some targeted therapies. We focus more on gene therapy, but the traditional gene therapy with adeno-associated viruses, but we are also exploring some other avenues, molecular therapies using siRNA, ASOs, etc. So the cornerstone of our studies is the human iPSCs, as I mentioned, from patients with genetic DCM that carry known pathogenic mutations. And over the past about a decade, we and others have developed two complementary approaches to study the disease in vitro. The first approach shown here on the left, we create iPSCs from patients with known pathogenic mutations. We're using also engineered nucleases, CRISPR, for example, to make isogenic lines. Meaning that we take each patient, we correct the mutation, and in the dish we have a control for each patient. That's kind of an important step in the process because studies have shown, and we have shown that the genetic background is a confounding factor, and comparing different patients, healthy versus controls, is not an ideal scenario. And here, what we do is having a more tractable model where we can compare directly the effect of a particular mutation. And we have developed protocols, we and others, to differentiate efficiently. Now the protocols are pretty straightforward. We can generate cardiomyocytes, and we do this in parallel for the controls and the isogenic controls and the patient lines. And what we do here is we mainly characterize disease phenotypes. So we compare the two phenotypes between the disease and the healthy equivalent. And what the idea here is that we can identify molecular targets, molecular pathways, and develop personalized therapies. So we have published extensively in this particular approach, and there's some references here if anyone is interested to look up, but I'm not going to spend too much time on this approach. But what we're going to do today is show you a complementary approach we developed a few years ago that instead of comparing phenotypes as we did previously, what we've done here is we focus on the rescue of a DCM phenotype using a phenotypic screening approach that combines functional assays, just showing here, for example, some contractility, calcium, electrophysiology. We combine those assays with a chemical screen, small molecules, so we're now expanding these chemical screens to more molecular screens using siRNA or CRISPRs. And the idea here is we can identify molecules or identify pathways that can rescue the disease phenotype, that can lead us to novel insights into the disease and developing targeted therapies. So for the first part, just to highlight some of the published work from led by Isaac, was a postdoc at the lab at the time. He moved on now to a senior scientist position at the local pharma companies. And this is the study we did a target discovery through the phenotypic screening using iPSC models and our high throughput systems. And the question we asked was, can phenotypic screening uncover new therapeutic targets for DCM? And to do that, we're showing here schematically, we use an automated contractility assay and a small library inhibitor of kinases. So what's showing here is the technology developed here at Stanford by Dr. Mark Mercola. So what we do here, we have a high throughput microscope. Essentially, we can see these cardiomyocytes in 384 web plates, showing here like schematically the cells spontaneously beat, and we can convert this spontaneously beating activity into a contractility kind of surrogate. So what we measure here is how well the cells contract, and when you add a particular perturbation, in this case a kinase inhibitor, what's the effect on contractility? And we have secondary assays to validate. We use a lot of engineered tissues, some microparent cardiomyocytes to measure the effect of particular molecules on the cardiomyocytes directly. And the goal is here to either discover small molecules, but also understanding the mechanism that the small molecules work, we can identify some novel targets. So what they're showing here is this is the screen of the 160 kind of diverse kinase inhibitors. These kinase inhibitors more or less target the entire kinome. What we found, just very kind of walk you through here, is this summary figure of the x-axis here shows the contractility, the amplitude, and here on the y-axis is its individual molecule. What you're showing here, anything above the dotted line here is a positive hit, means that it increases contractility as we plan to, and have some positive controls. This is omecamtiv mecarbil, one of the myosin activators and myosin inhibitors, negative control. So anything above the line is a positive hit, anything below is a negative, actually reduced contractility. Anything in between is actually has no effect compared to the vehicle control DMSO. So what we identify is two small molecules. One is this GO6976, which is a PKC inhibitor, mainly inhibits PKC, and the SP203580, which is a MAP kinase inhibitor. And we also did some psychodynamics, not shown here, everything's in the paper. We validated those in psychodynamic assays, but also combine this into one treatment. So we thought there's two different pathways. What happens if we put them together? And what we found actually was kind of surprising and exciting, because what we found here is that each one increases contractility as in the original screen, but when you combine them, you have this synergistic effect that essentially rescues the contractile deficit of these iPSC-derived cardiomyocytes, this mutant DCM patient-derived cardiomyocytes. And our control, when we corrected the mutation in this particular line, this is a troponin mutation, we actually see that the actual two molecule combination increases or rescues the contractility to a similar level as the genetic correction. So most importantly, what we found actually in additional assays, we tested this small molecule combination in several patients, shown here schematically, that carry mutations, these iPSC cells from patients that carry mutations on rare genes like phospholamban, common genes like titin, lamin A, tropomyosin, and LAMA2. So what we found that was kind of exciting is that regardless of the underlying genotype, these two small molecule combinations, labeled here as PPI, increases contractility in all the different lines that we tested. So if you have a mutation, these two combinations can rescue, at least partially, the contractile deficit of these patient-derived cardiomyocytes. I think the next question we asked is, what is the mechanism of action? We know that these molecules target two particular pathways, but we didn't understand exactly what the combination will do and what the underlying mechanism is. So we did a series of experiments showing here. Just one of the most informative experiments was RNA sequencing. We did RNA sequencing. We take several lines. We treat them with the small molecule combination, and we're shown here. We identify a very specific signature. I mean, typically when we did this experiment, when you add two small molecules, we have like hundreds or even thousands of genes changing in these cultures. In this particular case, it was kind of surprising. We only found about 130 genes were changing. And what was even more surprising, this particular signature was very specific to serum metabolism, glucose-related pathways, and amino acid transport. And what analysis shows that this particular signature is associated with the serine biosynthesis pathway. And serine biosynthesis pathway ranges from glycolysis, shown here schematically. It's a three-step process that produces serine, which is a non-essential amino acid, first catalyzed by the PGDH, the first rate-limiting enzyme. And through a stepwise process creates serine. Serine can be converted to glycine. Glycine is important in redox balance, or it can is a donor. Serine is a one carbon donor that participates in nucleotide synthesis and also in lipid biosynthesis and synthesis of sphingolipids. So it's kind of surprising because this pathway has never been implicated directly to any cardiovascular disease, cardiomyopathy, or DCM in particular. And it is extensively studied in cancer, and it has been implicated. Some mutations on some genes for the pathway have been implicated in some eye disease, including macular degeneration. So to confirm this molecular signature, we did carbon tracing. This is glucose tracing. And what we're showing here schematically in the graphs is that when you use this combination of small molecules in iPSC-derived cardiomyocytes from the DCM patients, we have increase in serine de novo synthesis that comes from glucose. Here we measure directly the serine that comes from glucose. The same also for glycine, confirming the molecular data. And what we've done, actually, one of the kind of definitive experiments we did, we blocked the PGDH with an siRNA to determine whether the serine pathway is essential for this beneficial effect we see with the two small molecules. What we're showing here, this is the control siRNA, we have the increase compared to the untreated cells or the vehicle-treated cells with SI. And when you block or downregulate knock down the PGDH, you abolish this beneficial effect, suggesting that serine is an essential mediator, or serine pathway is an essential mediator of the phenotypic rescue. So to summarize this part of the talk, and this is the published data. If you're interested, the paper has a lot more data there to showing how we identify and validated our target. And to summarize, what we're showing here is that the DCM iPSC cardiomyocyte phenotypic screening identified inhibitor kinase inhibitor combo that rescues contractility in a genotype agnostic fashion. And the mechanism we identified is the serine biosynthesis via the first enzyme in the pathway, the phosphoglycerin dehydrogenase. So for the next part, I'll just show some of our published and exciting data on our preclinical translation, led by two very talented postdocs in the lab, Mariam and Annie May. Annie May works also with Mark Del Capolo's lab, so we have a very close collaboration to develop these preclinical assays and test the potential therapies. So the question we asked is, can serine pathway activation protect the heart in vivo? Our in vitro data suggests that activating the pathway is cardioprotective. Now we want to take it a step further and see whether we can recapitulate that in vivo. So one of the first experiments we did, this is some of our published work with Timon Seeger from University of Heidelberg. So what we've done here is we did a functional screen to understand which point of the pathway is important in the phenotypic rescue. We did that because the pathway is very diverse and very complex, and there's, apart from the three-step process, the outputs of serine is diverse, as I mentioned earlier, including sphingolipids, nucleotides, and redox regulation. So what we've done here, we took a panel of lentivirus shRNA, a panel of lentiviruses to overexpress each gene that is important in the pathway, including the serine biosynthesis here, the three enzymes, one carbon metabolism, the sphingolipid synthesis, redox, and some of the transport, including the serine transportation. What is shown here is that PGDH overexpression in the DCM iPSC cells can rescue the contractile deficit, more or less equal what we originally observed with the two combo inhibitor. And that suggests that PGH was uniquely effective in restoring the contractility DCM iPSCs, and this is the key therapeutic kind of node in the pathway. We had some signal on the sphingolipids, but also in the transport, suggesting maybe transporting more serine could be also beneficial. But we focus mainly on the PGDH that we want to take forward into a more therapeutic target. So here's a hypothesis that enhancing PGDH-mediated serine biosynthesis cardiomyocytes in vivo is cardioprotective. To do that, we have developed a vector that expresses PGDH. This is an adeno-associated virus with serotype 9, which has a tropism in the heart. We deliver it systemically in the animals. In the humans, it's done intracoronary. And the idea here is that we can increase serine biosynthesis and we can cause this metabolic reprogramming in glucose utilization that could confer cardioprotection. So a proof of concept study to test this kind of hypothesis, we use a model. This is a tropomyosin 1 mutant mice. It's a transgenic mouse that expresses tropomyosin 1 alpha mutation, the E55K, which is associated with dilated cardiomyopathy. And what we found, and this has been a well-characterized mouse, this mouse does exhibit significant dilated phenotype as early as 6 weeks of age. So in here, our ejection fraction based on a 4D echo, about 6 weeks of age, we see a significant decrease, modest but significant decrease in ejection fraction compared to liter-mate wild type or non-transgenic controls. And what we've done is we have developed, as I mentioned earlier, this vector. We have delivered this vector at 6 weeks of age when you have the first signs of dysfunction, dilatation in this animal model, and we follow these mice for 10 weeks with serial echo, and at the end we do some tissue histology and metabolomics to corroborate the functional data. So what's important to note here is that we chose the 6 weeks because what we found was kind of surprising to some extent, because this gene wasn't associated before with the DCM, that at 6 weeks of age, there's no change in the gene expression. This is the mRNA. We see as the disease progresses, this correlates also with a decline in PGDH expression, both at mRNA level, but also in the protein level shown here. So the time we chose to treat these animals is to essentially prevent this decline in PGDH and see whether this correlates also with a stabilization of the functional phenotype. So showing here is just a summary of the data. This is, we also use the non-transgenic animal liter-mate controls to test if there are any toxicity associated with this particular treatment. As you can see here, the ejection fraction in animals, like healthy animals, treated either with a control, we use a GFP as a control, and our AAV9 PGDH doesn't have any effect on the ejection fraction of healthy animals. This is the control-treated animals, the mutant animals that develop this progressive dilatation starting at 6 weeks at the time we did the treatment and progressed to pretty severe decline in ejection fraction by the week 16 of age. But what we found very surprising and kind of exciting is that when we treat the animals at 6 weeks and we essentially prevent the decline of the PGDH expression, we see this stabilization of the functional phenotype. It's just stabilization of the ejection fraction, which by 10 weeks later after treatment, we've seen that the ejection fraction is more or less the same as they started. So what the data shows here, that preventing the deterioration or the decline of PGDH by augmenting its expression or supplementing its expression at week 6 can prevent the phenotypic decline in ejection fraction. And so here more quantitatively, there's a couple of images here how control-treated animal versus one of our PGDH-treated animals, you see you have a significant increase in contractility compared to the GFE. It's partially rescued, it never reaches the levels of the non-transgenic animals, and also that correlates with the significant decrease in this is systolic volume, but also anti-systolic volume between the two treatment groups, the controls and the PGDH-treated animals. So histologically, some examination showing here, this is our picroserous red staining with polarized microscopy. What we see quantitatively here is that the treatment prevents essentially the fibrosis in this animal model. You see this control-treated animals have a significant increase in fibrosis compared to the non-transgenic control animals. And PGDH decreases this fibrosis or prevents its development. And similarly, we see that the treatment also prevents hypertrophy. So here, this is WGA staining of the heart muscle, is the LV. You see the increase in hypertrophy in this animal model. and this is essentially is blunted or prevented upon treatment. And importantly, we didn't observe any toxicity. This is just histology, H&E, looking for inflammation or any signs of toxicity across the different groups. We haven't seen any adverse effect of the gene therapy itself in the heart muscle. So 1 second, my battery is running low. Okay. So to determine how this PGDH therapy alters cardiometabolism, we perform a series of metabolomics, both untargeted metabolomics and glucose tracings. And what's shown here, just again, just reminding the pathway is the conversion of glucose to serine. It is this pathway catalyzed initially by PGDH. And what we can measure here is both, we measured with untargeted metabolomics, both serine and glycine in the hearts of the animals at the 16-week time point, 10 weeks after treatment. We see here, at least in the cardiac muscle, we see a significant increase in serine in these hearts compared to the GFP3 controls. And we see also a trend a bit underpowered, but we see a trend also in increasing glycine, suggesting that the treatment does increase the bioavailability or the abundance of serine and glycine in the hearts of the treated animals compared to their GFP controls. And we did, more recently, we did some targeted metabolomics. This is glucose tracing. We have glucocarbon 13 glucose. We inject the animals after the treatment with, this is 16 weeks after. So 10 weeks after PGTA treatment, we fed the animals orally with 13 carbon glucose. And what's shown here is that the PGTA-treated hearts exhibit a significant reduction in the conversion of glucose to pyruvate, also and glucose to lactate. And together with the molecular and functional data, this shift suggests that restoring the PGT expression promotes metabolic reprogramming away from glycolysis and contributes to improved cardiac function in the DCM hearts. And showing here schematically, what we are proposing is that by overexpressing or restoring expression of PGDH, you can divert some of the carbons from the pyruvate to the serine to more biosynthetic pathways and conferring cardioprotection. I'm still working on the mechanism, but that's kind of the data shows so far that we can induce this metabolic reprogramming, at least from the glucose oxidation on the glycolysis part, and divert it to some cardioprotective pathways. Just for the final take-home message for this part, what we're showing here is that AAV9 mediated delivery of the PGDH in the TM54 DCM animals model halts the disease progression, reduces fibrosis, and prevents hypertrophy with no evidence of cardiotoxicity. And what we've shown is that this also correlates with increased serine levels in the heart that shows that the glucose metabolism has been rewired. And our conclusion, and this is what we've been working on the past few years, is that the AAV9 PGDH is a safe and effective metabolic gene therapy for DCM, and we hope to move this towards clinical translation. And with that, just going to thank several people from my lab. I mentioned a couple named Isaac, spearheaded most of the original discovery. There's Marian, so has been working with Anime from Michael Kapelov's lab on the translational part. Several people from Mark's lab that had developed the technologies that we use in the functional phenotypic screening. Snyder lab for providing several of these lines. We work very closely with Christian Metallo's group at Salk for the metabolomics, and Ariel did most of the metabolomics analysis I show you at the end. And we have an active collaboration with Tim on more of the genetic cardiomyopathies, but also on some of discovering some new targets. And of course, thank our funders. We've been supported from NIH for many years. Hopefully, we'll be continued to be supported by NIH for many years to come. The Duke Foundation, American Heart, have an internal program here at Stanford that we are working with to translate some of these findings into the clinic. And we have also support from several patient foundations, including the PLM Foundation. And with that, thank you for your attention. I'll happy to take any questions.
Great talk, Giannis. If you guys have any questions, any of the participants have any questions, please drop those in the Q&A. I'll start off as those start to trickle in. So have you measured any of the sphingolipids or any of these other, potential downstream pathways that you think might be kind of contributing, how increasing serine is actually invoking a protective effect?
I mean, that's a great question. Yes, we have tried and we still continue to try to measure both sphingolipids, amino acids. So what we found so far is just preliminary. Sphingolipid seems to be one of the major drivers, and we're still working on it. We haven't got, don't have the data to show you today, but we believe that sphingolipids might be one significant component why you have serine. Not necessarily making more, but serine and sphingolipids can be made also from alanine, and this is deoxysphingolipids. And Christian's lab has shown in other disease settings when you switch serine to alanine, you have this deoxy or toxic sphingolipids that accumulate, and he shows a recent paper that accumulation of deoxysphingolipids can cause peripheral neuropathy in diabetes. So we think along the same lines. It's a bit challenging in this model, I must say, because our therapy is directly to the cardiomyocytes. We need to develop some more assays directly to measure these metabolites in cardiomyocytes, which is kind of challenging when you dissociate the heart. So it is a bit challenging. So we're working with Christian's lab to answer this question. I think that's an important aspect that we don't understand yet, and I think we hope in the next couple of years we have better answers.
Okay, great.
Thank you for your question.
Yes, of course. So we've got a question from Navid Colini from Hopkins. I had a similar question as well, so this is great. I was wondering whether providing serine and diet would also rescue the phenotype. So have you tried any serine supplementations to see if you can circumnavigate the PHGDH downregulation?
That's a great question. I mean, we've had that question before, and we're working on it. I think what we found, is some preliminary data we're having, and as I mentioned, the serine can also be taken by diet. So we can supplement the diet with extra serine, and Christian's lab has done it for the peripheral neuropathy model. So we have done the same in DCM, and we see at least the preliminary data suggests that it's also a possibility that the serine itself in the diet taken up by cardiomyocytes might lead to cardioprotection. And I think that's one of the idea was that not necessarily develop a therapy here per se, but also identify particular targets. And I think supplementation is an easier experiment. I mean, when it comes up with some other challenges, but to answer the question, yes, we've seen our preliminary data suggests supplementation can also rescue the phenotype in this particular animal model.
Similar question along the same lines from Alan Titus. He's asking more about serine transporters, upregulation or downregulation potentially in DCM, or if that is also part of the mechanism of correction.
We have looked, we did some targeted approaches to look for the genes in the pathway when we overexpress PHGDH. There's some recent reports that also PHGDH goes to the nucleus and it has some transcription activity. It acts as a cofactor, a transcription factor. We haven't seen anything really beyond the metabolomics, but transcriptionally, we don't see much change in the transcriptome of the heart when we overexpress, when we supplement the PHGDH. So mostly is on the metabolic part, especially the glucose, like on the glycolysis and oxidation. I think what we've done, some of the screening we've done, as well as some of the data from Timon's work and Ramona's work, that if you increase the serine transporter, at least in the cardiomyocytes in vitro, we see some beneficial effect in terms of contractility. So I think there's another way to target this pathway. If you increase the transporter, theoretically, you increase the input essentially of the serine into the cardiomyocytes. That can be beneficial.
Okay. We've got a question from Dr. David Cass from Hopkins. How is the effect from PHGDH upregulation impacted by the extent to which glycolysis is activated? iPSCs are very glycolytic, so that may bias favor in PDGH, but larger adult mammalian cells are more lipid oxidizers and fatty acid oxidizers. And then also to kind of tail that off, what about in diabetes where this could be compromised?
Okay, so can you repeat the question so I don't see it?
Yes, I think the main
The two
question
parts, yes.
is,
What's the first question?
so iPSCs are very glycolytic, which would favor, a pH GDH side reaction. But larger adult mammalian cells are more lipid oxidizers. So how do you kind of convolute that potential implication?
I guess that's a fair question. I mean, our models correctly said that the IPS are more fetal, more glycolytic. And what we see more, at least from the PGD, the hard tissue that we've done metabolomics and the iPSC, we see a high activity of PGDH in the iPSCs. Whether these, I guess, if I understand the question correctly, whether these confounds our findings, I'm not really sure, because what we've shown actually in vivo, whether you have metabolic rewiring in these DCM hearts by kind of shifting some of the carbons towards the serine biosynthesis pathway or other biosynthetic pathways instead of the pyruvate, we do see some beneficial effect. So I'm not sure if I answered the question directly, but what was the second part?
And then just in the context of diabetes, have you looked at that?
We haven't looked at diabetes now, but in Christian's Metallo's lab, I mean, they've done some DVD mice, and I think they show that, I guess that the supplementation per se has a significant effect in function. This is, they're looking at peripheral neuropathy, but they haven't looked at the heart. So we're looking at other models, and we think that probably is more applicable to other metabolic diseases too that affect the heart. So this is work in progress. So hopefully we have some better answers down the road.
Okay, thanks. And then we have one more question from Raja Namakal Surapan. Great talk. Do you have iPSC cardiomyocytes for various laminin A mutations?
Laminin A.
Yes, LMNA.
Yeah. Yes, we do actually. We have a biobank here. If interested, you can reach out. There's a biobank at Stanford that has several patients derived iPSCs from different models, and including laminin A. Laminin A is one of the most common ones, about 10% of the patients, and we have several lines in the lab. And we're working. We have a Leduc Translationary Network funded recently to work on laminin A. So we are working actively on these areas. We have lines happy to share through our biobank here if anyone is interested, including the more of the lines that we have been using in these studies.
Great, thank you. All right, well, I think that answers all the questions. I'll turn it back over to Mark. Thank you very much, Yannis, for your time.
Thanks, guys. Thank you. Appreciate it.
Yeah, thank you to both of our speakers. Thank you to both of our moderators. Two excellent talks, great questions. We really enjoyed the session, so thank you all for joining us this afternoon, or perhaps morning, depending on where you're joining from. We're going to have another webinar we're planning for September, so please be on the lookout for an email from us regarding those future announcements. But again, please join me in thanking the speakers for their great presentations and the moderators for handling all the questions. So thank you again, and we look forward to seeing everybody again in a few months.
Thank you, guys.
Thanks, everybody.
Thanks.
Thanks, everyone.
Good day.
Bye.
Take care. Bye-bye.